Okay, so I'm very pleased that the final formal speaker of the day is going to be George Daley, the hero of the stem cell wars, and now the dean designate for the Harvard Medical School. He's going to talk to us about what to think about next, coming down the pike. Thank you.
Thank you. Well, it's a privilege to be able to try to give a bit of a summary and also to try to add some thoughts that might not have been provoked during the day. It was a really exciting set of talks from the folks who came before, and I do think that we have a lot to learn and a lot to contribute. I was on the plane down here this morning, as I'm thinking about the 6 a.m. flight, by the way, I was thinking about what to say, and I was reading an op-ed piece that Eric Lander wrote. Some of you may have seen it. It was published in the Globe yesterday, and in it he staked out a very fascinating position, especially for Eric. He asked the question, is the promise of genomic medicine overhyped? Question that he's been asked many times before, and he was asking it of himself. And his answer was quite characteristically, I think the answer is a clear yes and a resounding no. So Eric then had to resolve that paradox, and to do so he invoked something called Amara's law, who's articulated by this futurist who basically held that we tend to overestimate the effect of a technology in the short run, overestimate effective technology in the short run, and underestimate its effect in the long run. And I think the workshop today really provokes us to ask the same question of regenerative medicine. Is the promise of regenerative medicine overhyped? And I would answer that there is no doubt that is a clear yes, that it has been overhyped, but also a resounding no, which is that the opportunities for regenerative medicine to be transformative in the long run should really not be underestimated. So setting the right tone, and I think that's what we're really here to do, is to kind of set a tone for the future. It really requires you to take a bit of a historical note and to understand the timing of new medicine's development. And I've been thinking about this at various times. And if you go back to the advent of recombinant DNA, so if we trace that to, let's say, Cohen and Boyer in 1973, it's really quite remarkable to imagine that the first recombinant protein product that came to market was the Humulin by Eli Lilly in 1982, within 10 years. That was a remarkably fast translation. One could argue, however, that protein therapeutics as a consequence of recombinant DNA took more like 20 years to really come on. I mean, you had Epogen in 1989, and then you had a number of other blockbusters. Monoclonal antibodies, 1975, Milstein's paper, Kohler and Milstein. Within 11 years, Orthobiotech had OKT3 as a treatment on the market. It was 19 years before RiaPro, and really 20 to 30 years before we had blockbuster drugs of monoclonal antibodies, the Rituxans, the Herceptins, the many, many $1 billion of products that were the stuff of the late 1990s and 2000s. RNAi, 1998, Fire and Mellow. Still no product. And you may have heard a major setback for Alnylam. Still a lot of promise, but we're 18 years later. So what do we think about stem cells? Well, of course, you have to talk about the different categories of stem cells. We can argue in this forum, and as was presented, that we are enjoying the remarkable benefits of decades of investment in hematopoietic stem cell therapy and the merger of gene modification, the sort of recombinant DNA approaches to cellular modification, which we now see as gene therapy, which is really quite potent and effective in a whole variety of, of conditions. But when you think about other types of stem cells, and I think much of the hype and much of the attention has been around pluripotent stem cells. Embryonic stem cells were 1997. And we have clinical data. There's been clinical data on spinal cord injury. There's been clinical data on macular degeneration. There's maybe some hints of some early efficacy, but we don't have an approved product, and we're reminded of that by opponents of embryonic stem cell therapy very frequently. Embryonic stem cell therapy continues to be essential and important, as Lawrence Studer has proven to us, by choosing the first introduction in the Parkinson's work that we heard described, which is absolutely beautiful work, but it will happen with embryonic stem cells. So iPS cells, we fast forward another decade, so we're talking about 2007. And so we're now almost a decade from iPS cells, and we have had iPS cells in the clinic. We've had a patient treated in Japan and others on the way. But I think it raises the point that we're subject to irrational exuberance perhaps in the first decade, and we have to be reminded that technologies take 20 to 30 years to mature. And so what we've heard today is that we are on the cusp. If you think of it as kind of a wedge, we've got that advancing wedge of opportunity. We have remarkable platforms for thinking about treating neurologic disease with the products of pluripotent stem cells. We have remarkable data on beta cells, on RPE, on oligoprogenitors, on cardiomyocytes, an area which was not mentioned today, but will be in the news over the next week, is the remarkable progress in vitro gametogenesis and the interface between gamete production and the ability to treat fertility. So the bottlenecks, Lawrence really laid out, I think, a remarkably comprehensive set of issues around the bottlenecks that we face as a field of regenerative medicine. I want to put a highlight on the challenge of deriving the medically relevant cell types. It is a testimony to Lawrence's decades of investment in understanding the specific type of neuron and the developmental pathways that get to this specific regionally specified midbrain dopaminergic neuron that really casts him as the leader of the field and has put him in the privileged position to design clinical interventions which I think, from a scientific point of view, are highly justified. What he has done, and perhaps the best example, he has pharmaceuticalized a cell. He has defined a cell in such a way that you can assign it an identity. We presume you can assign it a potency, and he will be able to do highly controlled clinical trials that will define a dose-response relationship. And I think that's a very, very important lesson. In some of the other cell types, I think we are also approaching this similar pharmaceuticalized definition of identity and potentially potency. But there is no substitute for a deep and mechanistic understanding of the way cells work. I don't think I'm alone in the field of stem cell biology to feel a degree of inadequacy for our field when we use fuzzy definitions of cell identity and fuzzy definitions of cell function. I am not sure what it means to talk about the immunomodulatory properties of cells. Now, that is some sort of functional definition of their behavior in a number of immunologic assays, but it doesn't give us the kind of very rigorous, molecularly defined mechanisms. And why do we need those? We need those because that deep molecular understanding gives us the ability to assign a certain degree of confidence, a certain high probability that if we understand the mechanism, we will be able to predict efficacy, therapeutic efficacy. And even with that, we realize it's an imprecise science. So another issue which I think is kind of an overview of some of what I've heard today is trying to get an understanding of what is the right timing for clinical translation. And I think there's a risk to our field of premature clinical translation. Now, I'm not talking about the obvious risk of the sort of marketing of unproven interventions by the large number of stem cell clinics that are effectively operating outside of a regulatory milieu. I'm talking about the testing of weak clinical hypotheses that are coming through a organized regulatory framework. They're protocols that are vetted by institutional review boards. They are protocols that make their way into the clinic. They may be vetted in the context of a review by investors. But many of these are fundamentally empiric trials. Let's take a shot. We may learn something. And the problem is that they have a high probability of failure. And so testing weak clinical hypotheses leads to expensive failure. And we are drowning in the failures, and we're drowning in the expense. And fundamentally, everybody hears that it's too costly to develop new medicines. Well, we must reduce the regulatory burdens because the regulatory burden is so great. I'm going to look back on ourselves and say, why don't we hold ourselves to a higher standard of understanding at the preclinical level and take stronger hypotheses into the clinic? That's the way we're going to reduce the failure rate and reduce the risk. So I want to point out the risk isn't just to the patients. We obviously are concerned about not subjecting our patients to undue physical risk, but there's risk to the field for testing weak clinical hypotheses. So let me just finish by saying the International Society for Stem Cell Research, which I've been involved in for many years, has been attempting to be a steward of the field in part by really bringing together the community of professional scientists and increasingly clinicians who are involved in this space, trying to hold the field to the highest scientific standards, trying to promote communication, and also trying to set high aspirations by establishing guidelines. First, guidelines for the responsible conduct of what was a controversial area of stem cell research, embryonic stem cell research, but then later, in part driven by this rise of the sort of unproven medical interventions through stem cell clinics, to provide guidelines for clinical translation. We, after a 2-year effort by 25 scientists, ethicists, folks with some regulatory chops from 9 countries, we released an updated set of guidelines this past May. And let me just point out that was chaired by Jonathan Kimmelman, who's a really spectacular former biochemist, now turned bioethicist. And the core of the steering committee was folks like Nissim Benvenisti, Tim Caulfield, Helen Heslop, Insoo Hyun, Chuck Murray, Doug Sipp, Lauren Studer, Jeremy Sugarman, and myself. And there was a larger group that you can find on the website. But fundamentally, I invite you to read the guidelines, at least read the preamble. But what we tried to do was set a very high standard and a number of aspirational goals. And I point this out because lots of people wanted us to get much more concrete. But the point is, it's very difficult to get concrete when there's so much variability in every single cell type and every single disease offers a different scenario. So what we articulated were broad aspirational principles, and I want to outline those principles, and I hope you'll see and embrace these principles are a roadmap for going forward. So the core guiding principle, it was really an idealized process where peer review occurs through an independent, unconflicted, and expert review of clinical protocols to set a high standard so that we bring strong clinical hypotheses into testing. We articulated very high standards of safety, high standards of cell manufacturing and processing, standards that would be equivalent to and weighed against and balanced by the risks of the procedures, but also a very high standard of efficacy and mechanistic understanding as a precondition for clinical trials and ultimately the marketing of those therapies. And we spoke very strongly and collectively for a healthy skepticism about efforts to reduce the regulatory hurdles. You reduce regulatory hurdles at our own peril. We also pointed out, and I think it's extremely important to recognize, especially for a fledgling young field, the real potential for financial conflicts. And that we have to understand the financial conflicts of interest can corrupt the process of new product development. So we have to recognize when efforts to minimize regulatory standards are really being driven by financial and commercial considerations and aren't really in the best interest of our patients. And a case in point, and the ISSCR has taken a strong stand against the Regrow Act, which has been put forth in our Congress. It's being sold as a patient-focused effort to bring innovative new curative therapies to patients faster, when in fact it is reducing the regulatory burden in such a way that if the experience with cellular interventions is similar to our experience with pharmaceuticals, we know that if we're giving conditional approval on the basis of preliminary evidence of safety and efficacy, which is the standard of the Regrow Act, let's say early phase 2 data or preliminary phase 2 data, we know that the majority of drugs still fail after that. So should we be acting and enacting laws which actually require, essentially reimbursements for drugs which we know are more than likely to be either unsafe or ineffective? That's the kind of concerns that we raised in the ISSCR guidelines. So just read the preamble at least, and I think you'll see where we're trying to articulate the high standards. We are speaking to the integrity of the research enterprise, the primacy of patient welfare, the respect for research subjects, the essential need for transparency, and the aspirational goals for social justice. We really haven't talked about the thorny issues of social justice. Many of the diseases we talked about today, sickle cell anemia prime among them, is really a reflection of significant health disparities. And we in the stem cell community at least feel that if these new treatments are going to be developed, we also have to be paying attention that these novel technologies are going to be distributed and have access to those who can benefit. So I just want to conclude. I wish it were easy. I wish it were easier to make medicines that work. It's not easy. I wish it were less expensive. The path to creating transformative medicines is long and winding and difficult, and it is characterized by failure. We're not going to make it any easier, I worry, by taking the easy road forward. So I hope that this forum, by articulating the promise in a responsible manner, and I do think we're talking about that law up front, that even though we may have suffered from some irrational exuberance in the short term, we shouldn't lose sight of the fact that regenerative medicine is going to transform medicine. It is. It is going to be the medicine of the 21st century. You know, the 20th century, we learned to turn chemistry into new medicines, right? We now have to figure out how to turn cells into new medicines. And it's gonna take decades to do that. I think, and on behalf of the ISSCR, I would say that the best way to do that is to invest and to continue investing in the deepest scientific understanding of these processes. This is the way that we're going to sustain public interest to support the NIH. It's also going to sustain, ultimately, investor interest. If and when we can support a new company where the extent of preclinical evidence is such that we can carry forward with some confidence when we take it into the clinic. So I will leave it there. I think we walk a careful tightrope, because I do think we need to be speaking to the promise, but trying to give true hope and not false hope to patients. Thank you, and I hope I can take some questions afterwards.